All right, thanks everyone. Thanks, Kevin. I think I should have them with me enough whenever I get like the country sticker kind of setup you guys have here for this thing. So, okay, so let's just get right to it. Yes, I have not accounted for the 7 minutes that we're gonna lose. I was just amped that I'm gonna give a 15-minute talk instead of the usual 10. But I ended up again making too many slides, so I actually ended up running out of slides before I had to, before I actually got to the part that I was excited to share. So here it is. Here's my talk. So I will just talk like it's just a general overview of all the stuff we've been doing for the past years, and just like go through it. So hey, so my lab focuses on analyzing non-coding variation in genome, like how genome is regulated, and this is why we care about it. So this plot right here shows the growth of genetic information around here. So up there you have year 2000, down here you have around 2015, 2020, and then on this side you're seeing the growth of genetic loci associated with complex traits. So think diabetes, think cancer, that sort of stuff, Alzheimer's, whatnot. And so you see, like, there was nothing, and then around 2010, when we could actually sequence a lot of people, we started to get more, and now we're like kind of exploding out here. And this is one of the Mendelian disorders. So this is the type of stuff that you don't, most of the time you don't live long enough to reproduce. So they're pretty rare disease and they're pretty devastating generally. And so these things we already started picking up on them since 90s. They're way simple. They're not good diseases to have, but they're simpler from genetic perspective. There's like usually one mutation that gives you that. So there's like thousands of these that are known as well too. So this is really cool. Except that out of the hundreds of thousands that we have on this side, and about 10,000 that we have there, most of them are still unknown to us in the sense that out of probably 300,000 or so loci that are associated in GWAS. And so by that, I mean, let's say, for example, you would see things like that when you do studies. You say G to A transition on chromosome 10 position 8,000 blah 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 will increases your chances of getting PhD or decreases it. And that sort of thing is true. It's predictive. It's just not so informative and like somehow. There's like a pretty big leap. And the same thing goes with that, like when you have someone with a rare disease, like think a kid that obviously has a genetic problem, most of the time you just cannot tell what's wrong with them after genome sequencing. And the key to that is regulatory genome. So the idea is that these things probably affect the regulation of the genes, but we don't know how. And the reason it's hard is because the regulatory genome is pretty huge. So the non-coding part of the genome is almost all of the genome. And in contrast to the coding part, where it produces proteins, it doesn't have a clear alphabet. There are no codons. It's not conserved. If you look between different species, they're just like widely different from each other. And there are no direct products. Like the genes, you get an actual chemical molecule you can mess with and do things with. Here, you don't have an output. You don't even know what if you have a position here in the genome, what does it do? What gene it possibly affect? So that's what makes it hard. And so there are a lot of ways people look at it, and the favorite way that our lab works on it is like one of the most major ones is to use transcriptome data to do it. And out of transcriptome data, ETTL analysis is the most popular and established way to do it. So the idea here is that you're interested in understanding what does this, so let's say this is your genome, these are some genes there, and this is the area in between, and these little lines show the parts that there's variation in the population. So we got our first phenomenon for the talk, but there's 1 hour of this. So buckle up. So what you do to understand this thing is that you will go get about 100 people, a few hundred people, whatever you can afford, and then you do genome sequencing on them, you do RNA-seq on them, and then what you do is that for every position that is variable in the genome, you break people into 3 different genotype groups: homozygous, reference homozygous, all the heterozygous people. And you put their gene expression on these 3 box plots. And if you couldn't fit a line that is significant, you got an ETTL, that is an expression quantitative train flow list, which says this position of the genome is associated with the change of expression of this gene. So it's like the most standard way to do it. Now, another way that you could look at regulatory effects at a single individual is what's known as allospecific expression. And the idea here is that if you have one individual that is heterozygous for a regulatory variant, meaning that the two copies of the genome are carrying two different versions of that variant. So in this case, let's say it's this and that. Maybe this one binds pretty well to the transcription factor, this one kind of stops at it. What you do is that you sequence it, and if you happen to be lucky to have some sequence variation inside the transcript part of the gene, you could use that to identify that these reads with a C come from this copy, and these reads with a T come from that copy. And then you see that there is more of these ones than that one, suggesting that this copy has higher expression than that copy. Does that make sense? And if you do this for every gene in the individual, you get a cone like this, where each dot is one gene, and ideally, it will all fall on X equals to Y if your parents are the same person. And if your parents are different people, it kind of falls off of it because, usually that's the case. Now, there's the thing that is cool about this is that this signal, the allospecific signal, captures the net effect of all cis regulatory effects on a gene. And what that means is that it basically captures any effect on the genome that affects the actual physical molecule that variant is sitting on. So if you think of it, your chromosomes are not in pairs inside your cells. They're just lying around separately. So those effects that happen locally and not through some diffusible protein or something, they all be captured here, even if you don't know them. If you can't sequence this, it doesn't matter. The effect of it will still show up. And this kind of data has about 85% heritability, which is really good, meaning that most of the variation you see here is defined by your genetics. And that's because both of the copies of your chromosomes are going through the same cellular environment. It doesn't matter if I had a burger this morning and she had egg. It doesn't matter because I'm comparing myself to myself. But if you're looking at gene expression, that's a very different story. How old you are, what you ate, if you're sick or not, those things affect your gene expression. In fact, they describe 98% of your gene expression. Your gene expression has heritability of almost 0. It's not exactly 0, but it's 2%. So, cool. So, how do we use it? So, let's say now we have a non-nuclear thing. I'm going to probably get through about 20% on this slide, but let's see. Okay, so let's say we have the ATTL data. How does that help with what we have? So back to our problem with low educational attainment, and let's say this is the kind of plot we have. So in this spot, you have significance of each position of the genome in predicting your educational attainment. And so now if we have done ATTL analysis that we just taught for these same genes, so here we have the gene A and B. We looked at all the variants around them, and we actually found these two plots. So in here, I take this thing. So in here you have the significance pattern of genome regulating gene A and gene B. And if you look at this thing, you'll see that this one kind of looks like that effect, and this one kind of looks like nothing. So that kind of helps you to build this hypothesis that maybe that position in the genome affects the expression of gene A, and the change in gene expression of gene A in turn has something to do with lower educational attainment. Now that's not that helpful yet, but at least you have something tangible to go and mess with in a lab and find out how does that do. And this process of ascertaining that these two signals come from the same source is what's known as colocalization of ATTL and genomes. Okay. And another way that people can do this, and I'm not going to go through it, but I'm going to just put it here so that it's defined, is TWAS, or transcriptome-wide association set. The idea here in TWAS is that you cut the middleman. So instead of you doing ATTL analysis and then colocalize it, you just directly build a predictive model of genetically regulated gene expression, and you see if that's predictive of your trait. So does that make sense? Okay, cool. Okay, so the biggest consortium that has been doing things like this has been Gene-based consortium. So that's Gene Tissue Consortium, started about 3 years ago, wrapped up around 5. And basically this is a consortium that hired about 1,000 people post-mortem, but fresh. kind of like transplant grade 50 different tissues, and they did RNA sequencing of around 20,000 samples with whole genome and all of that, and basically systematically look for ATTLs across the body, and then try to basically use it to describe all the GWAS signal we see. So I'm not going to go through the details on this whole story, you can go read it, but the main takeaways that I want to share with you here is one, every gene has an ATTL. So before GTEx, we were thinking like, oh, if we find a gene with the ATTL, that's our hit. Turns out now, if you just sequence enough, you get ATTLs for everything. And two, the ATTLs are highly enriched for GWAS loci, 10, 20 folds, depending on how you count. But most of the GWAS loci after doing GTEx still are undescribed by the ATTLs. Okay. So the problem we had about 10 years ago when we were doing this GTEx stuff was like, how strong is an ATTL? So it's a relevant question in a lot of contexts. Like if you're like trying to look at the ATTL across different contexts, let's say different tissues, maybe different environment, maybe sickness, healthy, or maybe different ancestries, or maybe different type of assay. So somebody's looking at sequencing data, you have an NPRA assay or something based on qPCR. How would you compare these things together? Or you're interested in understanding how does these ATTL and ACA connect to each other? Like we know these are correlated, they make sense, but how? Or how much allelic imbalance is too much in case of, let's say, we'll talk about it, let's say, for example, the rare disease cases. So in that time, I came up with this exceptionally stupid model that did a track job. So as I said, gene expression is described as expression of a haplotype E of H is some factor Ki, positive factor Ki, times some basal expression ED. So the basal expression is everything that comes and is shared across two copies, and the K is everything that is associated with this specific variant. So we have Ki for this one and Kj for the other one. Okay. Now, this model, besides being very complicated, also involves two terms, neither of which we could actually quantify. So this is great. But the things that it does capture is that one, it captures the fact that we're looking at cis regulation, meaning that the effect, the expression on a given haplotype only depends on the genotype on that haplotype and not the other one. Okay? So it captures that structural constraint. The other thing it captures is that the effect is multiplicative. So the K here is multiplied to the basal expression, not summed or something like that. And that I'm not going to describe why it's a good idea, but it's a good idea and you can see it in real life. So how does this work? So I figured I don't have time to go through this, but if we write this for the simplest case where we have one variant, homozygous ref, homozygous alt, and the two heterozygous cases, and basically we just do that number for some two given case, you could basically see that in homozygous cases, you will have no allelic imbalance, you would have allelic imbalance in opposite directions in the heterozygous cases, and this would be the expression you get. And if you look at it in a population, this is how they will populate. The main takeaway you get from it that is the ratio of the two Ks is what determines how far these things stray off from the diagonal or how steep that slope is going to be on that linear regression. And the actual overall size of the case, it only determines the amplitude. And it's really actually something you can't quantify because it directly overlaps the library size if you're doing sequencing, for example. Or the amount of samples you collect or stuff like that. Now, so with that, we basically define the effect size specifically as the ratio between the K for the alternative and K for the reference. Now, we can't identify each of those Ks, but we could actually quantify the ratio. And we call that ratio allelic fold change. The interpretation of that is going to be the fold change associated with a copy of the genome carrying the alternative allele instead of the reference. Okay? This turns out to be also the same fold change between people that are homozygous alt and homozygous ref. Okay. Now, if you do some middle school math, you could use that little equation that we had to do some derivations about this. So, for example, you could show that the log AFC it actually has a lot of good mathematical properties, like it's symmetric and so on, it's additive, its absolute value is a metric, so it's actually really convenient to do a lot of algebra with it. Also, you could show that you could actually rearrange the expression for a gene that has one QTL to write it in terms of a linear regression. And in fact, the AFC in that case would be 2 times the slope divided by intercept plus 1 of that regression. So that gives us 2 things. One, that you can actually do linear regression to the QTL call, and 2, you could actually calculate the effect size of it from the parameters of that. And also, it could tell you that the log ASC is also the logic of the analytic ratio you observe in the ASC data. So the ratio of alternative allele to the total expression you observe, if you logic transform that, it also gives you the log ASC. And of course, naturally, the logistic transformation of the allelic ratio of the log ASC will tell you the log ratio that you will observe if someone carries that area. So that's like nice and convenient. And so we applied this to G-text. In here, we calculated the effect size based on that one from ASC of individuals that are heterozygous. And we calculated the effect size independently from PQTL data of individuals on that one. And if you see here, I don't see the correlation here, but it's about 80% correlated. And notably, it's not just correlated, it falls on the x equals to y line. So it basically shows that these two are not just correlated. Phenotypes are actually identifying the exact same phenomenon from the 2 independent forms of data. So, cool. So who cares? What's the big deal about overthinking what's the effect size of the linear regression? The thing that is important is that like the idea of what makes a difference in a certain space is a very fundamental concept about thinking about all things that happens there, in the sense that the AFC, as the way we define it, it gives you a formal framework to start now thinking about all sorts of more complicated problems concerning regulatory genomics in a way that you can actually know what you're doing. And we've been doing this over the years, so I'm not going to go through all these blocks, obviously, but I'm going to go through briefly 3 examples of how we expanded this thing into more complicated problems that we're interested in. So, 1, the most straightforward one, how does this generalize in case of invariance? 2, how can this be used to understand gray variant effects? And 3, how we can integrate other phenotypes in this. Tell you, I'm going to talk a lot about this, and actually does not involve the part that I'm going to talk about this.
Yeah, that's a question on the last slide.
Okay.
In the linear equation where we express log AXA on this 2 times are you putting the linear model on the original counts? Or is it
So if you actually, okay, yes and no. So when we published that, we gave 2 different ones. If you put in a log linear, which your actual noise would be approximately accounted for, then your ETL model would no longer be linear. So in that paper, we gave an approximation method that would do linear time approximation over the nonlinear model based on very stabilized model. But it turns out, also just do it on the normal non-transform data, you just basically, what is that, invert normalize the counts. It still works surprisingly well. Which is the description of what's in German known as Weltschmerz. And that's not when you realize that the world does not stand to the views of your thought. It just doesn't have all the stuff, oh, this is garbage, this is good, and then you do both of them, they're identical. So yeah, in the paper we did it both with and without lie. But yeah, if you put in log transformation, you could show that if the AFC is small enough, the model converges to a model, and the AFC would be 2 times the slope a lot bigger. Yeah. So okay, cool. Well, I'm gonna now keep you guys after because now this is it. Okay, so multiple layers. So at the time that we did this thing, we barely had any ETTLs. So at the time that we did, we only had 1 ETTL per gene. This was like GPEX was like the first major effort to do ETTL analysis. When the later versions came in, now we have every gene has multiple ETTLs. So these blue bars here is like the B8 version of this. It's the version that got published in 2020. Now B10 is out too. You see, this is the number of ETTLs per gene. A lot of genes have more than 1, and this is only in 1 tissue. If you actually account for all multiple tissues, every gene has many ETTLs. So, not going to go through how you find it, but at the time that the paper was published in 2020, they just went ahead and used AFC to calculate AFC as I just described before for a single variant to calculate the effect size of all these conditionally independent BTTLs. So these are BTTLs that are still have an effect size after you have marginalized out the previous findings. So that is this model, right? So you basically have, for each BTTL, you assume the same model. You basically try to estimate this effect size, given the stuff that is said. The problem with this is that it's completely ignores the LD structure, and it's also agnostic to haplotype effects. So I have a slide about the haplotype effects that I briefly will show it to you guys. With the LD issue, I don't have a slide on it here, but basically the issue is that if you do this, if you have more than one variant, if you can imagine there's a bunch of variants that are affecting gene expression. In an IID scenario, you could just do one at a time and ignore it. The fact that you ignore it just confuses your variants, right? But in the real world of human biology, about every variant is in high LD, meaning that they're not randomly distributed, they are always correlated. So now if you have a secondary variant with a different effect depending on how correlated they are, you're systematically biasing your estimates. Now with the haplotype effects, is this the case? This is the case. So imagine you have two variants here. The red one is the high expressed one, the blue one is the low expressed one. Depending on if the two high ones and two low ones sit on the same haplotype, or they sit in the flipped scenario, you would expect obviously to have a very different allelic balance. But also you expect under this model to have a very different expression level. Right? It's very easy to imagine this in an extreme case where your low expressed variant has zero effect. Right? Like it just kills the expression. In this case, you kill both haplotypes. In this case, you kill only one haplotype. Right? And so this is what the model tells us. This is says the difference between these two expression of these two conformations of the haplotype is going to be this. So this is the effect size of each one, and that's the redder it is, the more these two are diverging from each other. And if you actually look at the real data from GTEx, you see that now this is each dot is each individual. You see that you get the exact same pattern shown up. So these capture when you do them one at a time. And indeed, when you run I forgot to. So, anyways, we had this model, which is just a just simple extension of AFC model to the invariants, which is simply the sum of their effect sizes, and then you could just do a vector multiplication to get like the expression for a setup where you have invariants like this. So if you apply that, you will get the situation. So here you have the genes with 1, 2, 3, and up to 6 and more ATTLs. And here we have on the y-axis the described variants by the ASTM in the blue and the original AFC that was used by final release in red. And towards this end, you're describing almost twice as much variance with the ASTM model. And if you actually use the ASTM to predict gene expression, so this is important, this is something you use in TWAS, it actually performs very comparable to SUSY and ElasticNet. It's significant, it's statistically significant, but it's within the same order of magnitude. So it works great, and it produces LE-specific predictions as well. So for those that might be interested, we have already produced genetic regulated gene expression for both haplotypes of all UK biobank people. So okay, I'm going to stop with that one. I'm going to get into the detail. Now I'm going to talk a little bit about the case 2. So the case 2 that I want to talk about is the case of identifying rare variants that have large regulatory effects. So why do we care? Here's a motivating example. Rare diseases. We talked about it. There are thousands of them. They're devastating diseases. And usually you do sequencing, like whole genome sequencing of the individual and the parents, siblings, whatever it is there. And they can look for smoking gun cases. Smoking gun is a case where you have a gene like this that does not tolerate any variants in a population. It's associated with your disease. Let's say it's a brain gene, you have it looking at the brain symptom, and then you happen to have a stop gate in it, or a frame shift, or some sort of protein truncating there. That's a smoking gun. That is a diagnosis. Usually, you'll get that kind of thing less than half the times. After that, you're left with no diagnosis. You know something is wrong, but you can't go on. And that's because as long as you don't have that sort of type of obvious coding mutation, you can't say what's wrong. So the idea was, let's just look at, now that we know the gene is not destroyed, let's see if the gene is dysregulated. So the idea is here. So we have genotypes, we have a lot of people. This is our proband. This is the person that, this is the individual that is sick. We look at every gene, we look at the distribution of expression for that gene, and we see if this individual is an outlier with respect to our reference of genes. So that sounds great, except that it just doesn't work at all, right? Your proband, you're looking at maybe the 2-month-old that has, let's say, muscular dystrophy. This distribution comes from a set of post-mortem 70-year-old SAM individuals. So whoever has done gene expression analysis and understands what 2% variability in gene expression mean, would know that this is just offensive to them. So you can't do that, but the idea is key. So the alternative way that we can, it's not like, it's something other people have done before, is the idea of like, why not use the program as a both case and control? And the idea here is that most of these mobility disorders, at least the ones that we know, we know they're dominant. You're looking for one mutation. Okay, so you know that the person was okay, they've got one mutation or one copy of the genome, it's heterozygous. So the other copy is perfect. So let's just use that as the control and the other one as the case. So the idea is here. So you basically do RNA sequencing, you do produce ASC data for each gene. Now you produce it for every gene. You do your favorite statistical test to identify genes that are out of whack. And they basically say, hey, look at this one. This one is so off balance. Probably something went haywire on one of the copies. That's why it spills so out of it. And so the favorite ways to do this kind of test is going to be binomial, or if you're fancy, maybe beta binomial, some other form of like over dispersed test. The cool thing is that the ASC is going to be super robust because both of these are inherently controlled with each other. And the other thing that is cool about ASC is that it also captures the cis effect of every variant that is out there. So even if the variant that is causing this imbalance is something you just can't sequence well, maybe it's some weird like part of the genome that is just hard to sequence, you still can see its effect here. So that's really nice. The thing that is nice, not so nice, is that it really doesn't care about all these hundreds of millions of dollars that are going into GPAX or any other public effort to produce all these reference data sets. So it just, every sample is just treated like we've never seen this before. So how do we go about this? So one idea is that, well, this is our patient sample for each gene. We go to GPAX. We go like, this is my gene in my patient. Let's just look at the ASC of this gene in every individual in GPAX. And if this thing now falls out here, bingo, you got yourself ahead. The problem is that if you go to GPAX, you might fail in any of these scenarios. In fact, when you look at ASC across GPAX, it looks all over the place. They have all sorts of patterns that are not necessarily so trivial and difficult to describe. And the reason behind it is that there are a bunch of regulatory effects in a population. We're not equal people. We're equal. Well, not so equal. Equal are different. That's it. So we are naturally different, and we're not different in a uniform way. We're not some petri dish people blended and go like, you two be parent of this one, you two be parent of that one, right? There's all these very clear structures across the human population that makes these sort of non-random variation that have such clear patterns. And then to make matters worse, when you're looking at ASC data, you're seeing everything through the lens of the variant that happen to be in your coding region. That's the proxy that helps you measure the regulatory effects, and that also biases everything. So how can we compare all these things in a unified way? So this is the strategy we took. So the idea was like, let's first characterize the distribution of regulatory effects for each gene. Loosely speaking, that is like, let's find out all the variants in the population and their frequency and effect sizes. So if we know all of that, then we could estimate, then we could estimate the amount of genetically driven genetic dosage variation in a gene. So we can find out, loosely speaking, how much of gene accepts being messed with its regulation in a natural population. Like how much variation is okay, the same way as with height. If you see someone is 5.5 feet, you're like, well, there's something off with you. But if you see someone is 3.5 feet, you're like, well, that's outside of the normal range of height difference, right? And so this is kind of, yes, like what is that normal range for each thing? And then finally, when we have that thing, we can come up with a hypothesis test that basically sees if this gene is out of that distribution. So how do we do it? Well, we know how to do it for the case of one variant in the population. So if you have one variant in the population from the AXP groups, you could basically say, hey, in this case, we're going to have three groups. The three groups is going to be homozygous ref and homozygous alt. Both of them are going to have no allelic imbalance and going to sit in the middle. And then we're going to have two groups on two sides corresponding to heterozygous, where the case that I express one on the left side and I express one on the other side. Okay. And the actual allelic ratios for them comes with the logistic transformation of the effect size of this variant, right? And how many individuals fall in each of these groups? That also, I'm not going to go through this, but it's basically simple multiplication of how many times do you expect to see this guy at the same position as this one, and how many times do you expect to see this one now? So it's defined by Galilean frequency and delta, not complicated. And now if you want to take into account the fact that this is count data, it will be a three component binomial mixture, okay? Now, if we take it to the case that is probably in reality where we have many variants, then this thing will turn into slightly more complicated, but not that complicated. It's basically, you will expect to see now a binomial mixture with 3 to the power of n components. Basically, every variant will kind of branch it into another 3. And each of those components, the ratio, like where they fall in the reference ratio, is going to be defined by the sum of the effect of those variants that are there, logistic transfer. And then the frequency is going to be simply the probabilities multiplied each other. Not complicated to write down, because that's what's good about math. In practice, on the other hand, obviously everyone understands this is just not a method you could actually fit. We will never be able to actually make this model work. So thought of a little simpler scenario where you actually do know only a subset of these variants. So let's say we have n variants, but we only know about m of them. And then the rest of them, they're unknown to us, or somehow we don't want to explicitly model them. How will things change then? Well, in this case, when you have only m of them, only m of those would come into the group's frequencies, because now you only have 3 to the power of m. So these number of groups you see shrink to only m, to the genotypes you're directly copying, and then the rest of it will get captured in how fat these things are going to become, right? So everything else that you're not modeling explicitly here is going to come out of this sum to a separate sum of the effect sizes, except that in this case, it's not just directly the sum of the effect sizes, it's the expectation of the effect sizes as defined by their effect multiplied by their velocity problem. Okay? So now if you do that, under reasonable assumptions, you could assume that this sum is going to be described by a normal distribution of some unknown variance in 0 B. So now, if you get this logic on the other side, you would say that logic transformed reference ratio is going to be normal distributed with a mean defined by all variant effects that I know, and then a variance associated with the rest of the variant that we're not accounting for. And these two are simply even variants of log A of C. That's their interpretation. Now, the thing that is cool is that this is just definition of logic normal distribution. And it just happens here. So basically, the true distribution underlying each of these components is going to be logic normal distributed with the mean and variance defined here. Okay. Now, if you add the fact that there's a binomial noise added on top of it, then this will become a binomial logic normal mixture model that is defined by basically effect sizes of the variance you want to explicitly model, and their allele frequencies and their LD to these things, and the net effect of everything else that you're putting out in the model. So that's easy enough to fit. Now to do that, so we could fit this thing, it's easy enough to fit. Now that we fit it, it's the next step. So now we have a distribution that basically collects sufficient statistic of everything that is happening in a population and can model all of these within the same framework. Then all you need to do is to calculate the expectation of dosage variation from the standard variation model. So now in this case, these are going to be little like they're going to come from these integrals. So it's a little more complicated than this, but nothing really that you cannot do easily with a numerical integration. So now that we have everything, so we have our population, use that population to estimate the genetic variation in dosage. Now that we have that, we can set up a little binomial test that is over dispersed to account for a variance to see basically for each individual gene, can this amount of LLVT balance come from a variant that can throw it dosage off by more than this or by this? So that's the normal. So applying this thing to a data set of 70 individuals with muscular dystrophy that were previously diagnosed, this is how it looks like. So this is one of the patients. This is AST data for it. Each dot is a different gene. This guy in green is the gene that is actually causing the disease. We know experiment. If we do a binomial testing on it, we'll identify every gene that is statistically out of balance and catch it. The problem is that we also catch 407 other genes. That's not so much. If we do a beta binomial, we end up doing the cut a little further out, so we only catch 66 genes. But then we miss this guy because it's not all that far from the area. Now, if we do anything with that, we'll actually find 19 genes and we catch this guy. And meanwhile, we don't catch all these other genes that are way out there because we know they're just nice, lovely genes. Now, if you do this for every individual and compare it to a bunch of different methods that we decided or the universe forced us to put it in there, we want to be here. If you were God, you would be sitting right here. You would report one gene at 100%. So we didn't quite make it, but we are pretty high up here. So we could find everything that you could capture with a binomial test, which would be the gold standard for sensitivity. But we found a lot less. We found on average 11 outliers per individual and caught everything that could be theoretically caught. So it was great. Now, applying this into a similar case of population testing. So that was tested for our population. You learn from genetics data, you apply to data set of pediatric diseases. In this case, we're actually just going to apply genetics data to identify individuals that carry rare variants. So that's very important because actually all of us carry millions of rare variants. Statistics are useless for analyzing them because we just don't have statistics on them. They're rare. That's the idea. But they're very important. It turns out all of these complex diseases, like diabetes and cancer, all these things that are really popular, you find in GWAS, they also have a big component of rare variants. So you can have rare variants that you have it, or your family has it, no one else does, that they have far larger effect than the stuff that are found by GWAS, but we just don't know how to use them. So they're kind of very important for precision medicine. So now we apply this thing to genetics data, and so this is the enrichment of in genes with a variant that is rare and is likely causing serious effects like splice variants or stockings, and we see you produce around 30, 40, sometimes 100, depending on how you cut it, enrichment for genes that are affected by these rare variants. And this is like many folds higher than the other methods can do. So this is like the yellow and this purple are the two different varieties that you got here. And so, so here's back we banded with Mohammadi-Stat on Stephen Montgomery's lab to now run this systematically with also outliers for gene expression. We Stephen's lab did and splicing where Alexis lab did, and basically systematically look for variants of large effect in genetics data and build a little Bayesian model to basically use the transcriptome data to help improving interpretation of these. So with that, I'm going to stop on that part and use the remaining time I have to talk a little bit about integrating other phenotypes. And so this is the idea. So a few years ago, I was just thinking like there's just so many of these things we can do with gene expression data. So we get the gene expression data, and then we analyze it usually for gene expression. In gene text, we also use the narrow sense definition of splicing. So this is leaf cutter, which looks at the intron excision, which is far from the entire story of splicing, but it's a big part of it. And then we use these things for a lot of things like TTL analysis from GWAS, TWAS, PWAS, heritability. Like there's a bunch of different analysis that people do in these things. You could also use it for outlier detection, differential analysis, all these. And so that sounds great, but the thing is that we also know that RNA-seq data, that includes mRNA-seq data, actually is known to include a ton of other things you could measure from it that we know. There are individual papers that have shown that these things actually find biologically relevant things, like isoform usage, link RNA expression, circular RNAs, RNA editing, alternative transcription stop site enzymes, a bunch of other phenotypes that you could measure in your RNA-seq data, and we know they're relevant, but no one ever uses them because they're just a pain to incorporate. So a few years ago, we came up with the idea of like, can we just implement a bunch of these things in a unified system that can just do it, and we can just already move on as humanity from this thing and just like do it. So this was the first shot that we picked. This is Dan from my lab and the Mom's lab that did this. So the first version of the implemented expression isoform ratios, intron excision rates, alternative poly, alternative TSS, and RNA stability unit. And basically build a unified method that you could add more modes to it. And then the idea is that if you just throw the transcriptome data at it, it produces all these phenotypes in a consistent fashion for it. And then if you want it, it can also produce QTL data, TLOS, and co-localization, all downstream analysis, so all of them consistently. And the thing that's cool about it, it also analyzes these things jointly, so it can account for correlations. That's one problem with adding more of these things one by one, is that most of these things are heavily correlated with each other. They produce confounding signal that just becomes completely out of control. So, okay, so how does this look like? When you run it, this is, I think, in GTEx or Geotics data. So this is the number of QTLs we find from each of these phenotypes. So you see the biggest one by far is the gene expression, because it's the most powered one. But also we find all these other QTLs in other phenotypes. And we find more than twice more QTLs actually when we look at these other phenotypes. And when we look at them, so this is like a recursive finding of the independent signals. And this is the first QTL find for a gene. This is the 10th one. You see that from the beginning to the end, at the beginning you get a lot of expression ones. As you go further, you keep on getting more and more of the other ones. But basically you have a distribution of different things. And when you look at the variants that underlie each of these QTLs, you will see that the variants that make expression QTLs are heavily biased toward the promoter, a little bit towards the end. The splicing ones fall mainly inside the gene body. All TSS, very dead focus on the TSS. All polyase focus on the end, and stability goes all over. So you actually come in from different parts of the genome. They're actually dependent signals that come out. And so when you use these things to interpret GWAS, this is obviously a cherry-picked example. You could get cases like this. So this is like a gene for neuroticism. And so in here you have the GWAS signal for that gene. And you see here that in isoform ratio for one of the isoforms of this gene, you see a signal mirroring this GWAS hit. But actually, when you look at the expression, alternative TSS RNA stability, you see nothing going on. And if you do this systematically for everything across about 100 different GWAS data sets, this is what you find. So these are each one of these lines is a different phenotype. So on top, you have height as defined by UKIP biobank. You see that we found about 800 different co-localized hits, and these are independent hits. from each other. About a third of those come from expression, about 2/3 of them come from the rest of the phenotypes. So that's pretty cool. Basically improving stuff quite a bit. So that's cool. There are a few problems with this. So one is that these different modalities that we find are actually very correlated to each other. So analyzing it is not straightforward. So we've kind of done it with some, duct tape or chicken wire, but like it's not straightforward to analyze. Huh? And so incorporating new things is always a pain. It's not straightforward for most reasons. And what if there's more of them? What if we stop? So what we were looking was like, can we just do phenotyping of gene expression in a data-driven fashion? So the way we did it is the first version we tried is that you get the genome, you segment it with your favorite form of segmentation. I'm not going to get into how we do it. And then you go look at a population and you see what are the patterns of change in those segments of the genome when you're looking at the coverage of RNA-seq. Simplest way to do it is to do PCA. We looked at PCA, functional PCA, a couple of different things. And then you use the latent dimensions of variation as you find the population as the phenotype. So you say there seem to be these forms of variation across population. So this is how it looks like for a gene. So this is the gene. These are the transcript isoforms that are available for this. Mind you, the method actually does not use the annotations. So it doesn't know about these. This is the coverage you see on RNA-seq for this gene. And this is, for example, one of the components we identify. So the red ones are people that are sitting at the top decile, the blue is the ones that are below decile. So you see this component captures this kind of variation where people have higher stuff here and lower stuff there versus people that have higher stuff here, lower stuff here. Now, if you plot these, and we call them data-driven phenotypes versus knowledge-driven phenotypes, so expression, isoform ratio number one, blah, 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 TSS, all poly AR, and stability, you see that the first component we found is almost perfectly correlated with gene expression. You can imagine, gene expression is just the sum of the effect, the sum of the coverage. The second one is heavily correlated with alt poly A phenotypes. This seventh one is correlated with this, and so on. And then some of them are just not correlated to any of these things we know about. So now the thing is that you could just use these things just the same way as you use genes in QTL analysis and TWAS and all of that. You could associate genes with phenotypes to them. And so this is what we did here. So this is all the results we get from 6 phenotypes implemented in Pantry. This is when we just use these phenotypes we got from this data-driven phenotyping. You see in here, we got about 22,000 ETTLs. In this case, we get about 30,000, like improving like 30%. Now, obviously, these things are much harder to interpret than these guys. So, of course, we could also do them after residualizing these things that we know about. So in this case, we first residualize everything we know out of our variation, and then we look for data-driven phenotypes after that. And now we get 30-something thousand, that is like 40-something% more QTLs there. And meanwhile, we still retain everything that was interpretable. Now, if you use these things to interpret GWAS data, in this case, in the white, I'm showing everything you can identify with these 6 phenotypes. The gray one is the ones that you identify with these phenotypes, but also you identify with these phenotypes. And in black are all these new ones that you could have never found. So you actually improve the GWAS loci that you described also by another 30% on top of what we could do with Pantry. So the take home, hey, look at that. Okay, so the take home is, so I just gave you a general lecture about use of transcriptome for understanding genomics. I hope we not taken a course on that, but if you did, it's always a pleasure to listen to things you already know, makes you feel smart. And I talked about knowledgeable J, which I believe that's a good way to think about gene regulation. But also that's like generally the way I want to think about any problem I want to model. It's very important to just take a moment to think, what is it at the heart of the problem I'm trying to tackle, and understand what it means for a change in that field, and then based on that, you can build an entire framework to actually help you build more complex cases that otherwise you can make crazy choices. And talk about transcriptome assisted variant interpretation, and then use of multimodal analysis of RNA-seq data. If you're using RNA-seq data in your work, this just increases your finding impact of it. At no cost, these things are available. We have them implemented for Nextflow and whatever flow things you kids use. So it's good. Use it, it's good for you. In that. So future outlooks, where are we going from this? So one of the things that is exceptionally exciting about these data-driven phenotypes is that they really don't depend on that annotation, which is really nice because the annotations are kind of a bottleneck. And so every time we do long read sequencing of RNA-seq, you find that, oh, there's like majority of the things you find you've never seen and they're not patterns. So these don't depend on that. So it makes them really exciting to use them for rare variants because it's reasonable to assume rare variants can cause things that are not happening, right? But the problem with them is that they are not magically possible. So they really have lack statistical power to actually look at things across genome the way we do when we do rare variant analysis with splice. So one of the things we're doing, we've hired one of the finest minds of this department, a young man, and he is working to basically combine CCA and fine mapping and functional annotations all into one big model that tries to learn these patterns across all genome with respect to functional and matrix that they have to identify latent phenotypes that are actually consistent across genome and to use them for rare analysis. And I think if you're interested, this is the part that I want to talk about, but obviously I'm not, is the part that also another way to actually incorporate all these different phenotypes into our analysis is to actually try to fuse data not by your generic data fusion techniques like CCA, but use it by taking advantage of the inherent links in your data. So think about how your data is inherently connected and use that to basically mix them into each other. So here is kind of an overall sketch of how isoform expression and stability and composition and all of that is connected to the gene expression and the variants that are in there and how we're used. So this is something that I've been working on for the past 3 months and I'm very excited about. I'm not going to talk about it, but the loose idea is that we're borrowing some ideas from computational data analysis to basically make a uniform model that can take into account gene expression effects and effects that change the composition of isoforms in a gene, all into one unified model, and basically be able to not only quantify them but use them all systematically to us in a way that they're not redundant with each other. And the cool idea is that it turns out the AFC model and everything I said from single variant to multivariant to the NUDA applies exactly the same way if you consider transform methods from computational data analysis. So hopefully next year I'll have better results to describe on that part. But with that, I'd like to give you a long list of people that have been born in the politics. And with that, I'd love to thank you, and I'm 1 minute over time.
Thank you. Thank you. Any takers for question? Maybe I can ask one. I might have asked one how this at some point. How pervasive are allele imbalances like across like cell types or diseases? Like, should I be thinking this is like literally everywhere and I just was none the wiser, or is it like more common in certain cells and certain diseases, or
So
is that a sense of scale?
no, so you saw like the binomial plot I had with the binomial test. If you just look at your average RNA-seq data, you have around 400 or 500 genes that they legally are in allelic imbalance, meaning that if you do a binomial test against 50-50, they're off of that. It's not necessarily identical across different tissues. It's not necessarily identical across different contexts. So for example, you could have a gene that is in balance under homeostatic conditions, but if you have flu, one copy starts to behave differently from the other.
I see.
So it's just like really mirrors the regulatory variation in genes. So if you go look at, I don't know, P53 or some sort of gene that does not like to mess with like that, they don't have usually large effect EQTLs, and they do not usually have LSC expression. That said, you could have genes that usually don't have EQTLs because they're very intolerant, but an individual might carry a rare variant that would make that gene to happen to have alpha-specific expression. Okay.
Anyone else?
Just nailed it.
Maybe I could ask a
Well,
question.
maybe we can talk about random other things, but let's first thank our speaker one last time.
Thank you.
And hope to see you all next week.
Oh, Ali had a question.
I did, but
Hey,
looks
Ali.
like, you guys hear me?
Yes, I think.
Do you hear me?
Yes.
Okay. Sorry. I mean, I could ask Pejman later. I mean, if everyone's leaving, that's okay. I could ask him later.
Yeah. Okay. Email me.
Okay, we'll do.
All the best.
Yeah, thank you. Nice talk. Thank you.
Thank you.